Welcome to the deep dive, where we cut through the noise to bring you the insights that truly matter. Today, we're diving into the microscopic world, looking at prokaryotes and viruses. This is all based on Chapter 13 from Raven Biology of Plants. Now, you might think of these as, the smallest players on the biological stage, but as we're about to discover, their impact, well, it's anything but small. Our mission today is to really distill the core knowledge from this chapter, give you a clear, engaging understanding of these fundamental organisms and infectious particles without getting totally bogged down in detail. Okay, let's unpack this.
Yeah, and what's truly captivating here is how these tiny entities, from the very first forms of life on Earth right up to the invisible agents causing devastating diseases, they're absolutely everywhere. They're constantly shaping ecosystems, even our own bodies, in really profound ways, often surprising ways too. So we're going to explore their unique structures, their ingenious survival strategies, how they reproduce, and their critical interactions with the world around them. By the end of this deep dive, you should have a solid grasp of why these microscopic powerhouses are just so incredibly important, even if you can't see them with the naked eye.
Okay, let's begin with prokaryotes then. They're often described as the smallest and simplest organisms. But simple, that can be a little misleading, right? Here's where it gets really interesting. Despite their microscopic size, their collective weight on Earth, it's estimated to exceed all other living organisms combined.
It's staggering, isn't it?
Yeah. Imagine they make up 90% or more of living organisms in the sea by weight. And a single gram of fertile soil can hold billions of individuals. Plus, they're incredibly ancient. We're talking fossils dating back 3.5 billion years. That ancient lineage and just the sheer abundance, it suggests an unparalleled mastery of survival.
Absolutely. Their incredible success really stems from their extreme adaptability and rapid reproduction. Take E. coli, for instance. Under ideal conditions, it can double its population every 20 minutes.
20 minutes?
Yeah. This allows them to quickly colonize and thrive in environments where pretty much nothing else can survive. Think icy wastes of Antarctica, near boiling hot springs, oxygen-depleted deep-sea vents.
Yeah.
They're there. And it's crucial to remember there are two distinct evolutionary branches here, Bacteria and Archaea.
Right. Both prokaryotic, but very different.
Exactly. They are as genetically distinct from each other as either one is from us eukaryotes. And then, completely separate, you have viruses. They aren't even true cells. They're basically just genetic material that needs a host to replicate.
So if they're so simple, what does a prokaryotic cell actually look like on the inside? No nucleus, no other specialized compartments. What's going on in there?
Well, instead of a distinct nucleus like our cells have, they have a single, often circular, DNA molecule. It's supercoiled, packed really tightly, but it's localized in a region called the nucleoid.
So a defined area, just no membrane around it.
Precisely. And this DNA can be surprisingly long, thousands of times longer than the cell itself, just twisted up compactly. They might also carry smaller, independent loops of DNA called plasmids. These often carry useful genetic traits like, say, antibiotic resistance.
Right.
And while they lack those membrane-bound organelles we have, their internal space isn't just, empty soup. Many contain tiny protein-shelled compartments filled with specific enzymes. Plus, you'd find thousands of ribosomes, the protein factories, and distinct stored granules for carbon and energy. Some, like the photosynthetic cyanobacteria, even have extensive internal membrane systems called thylakoids, where they actually capture light energy.
Okay, interesting. And what about their outer layers? What defines them from the outside?
Well, they have a plasma membrane, a fatty layer, much like ours, controlling what goes in and out. Crucially, it's also where key processes like respiration happen, and sometimes photosynthesis too. Most prokaryotes also have a rigid cell wall outside the membrane. This gives them shape and protection.
And that's where the peptidoglycan comes in for bacteria.
Exactly. In bacteria, this wall contains a unique substance called peptidoglycan. It's so unique it's considered a signature molecule. And scientists use a staining process, gram staining, based on this wall structure. Gram-positive bacteria have a thick peptidoglycan layer that holds the dye, making them look purple. Gram-negative bacteria have a thinner layer, plus an outer membrane so they don't hold the dye, and usually appear pink or red after counterstaining. This difference is actually really important in medicine, for instance.
Makes sense.
And on top of all that, many secrete a slimy outer layer, a glycocalyx or capsule. This helps them stick to surfaces, resist drying out, and sometimes even hide from host immune systems during an infection.
Okay, so they're well protected. Beyond these layers, how do these tiny organisms actually, get around or interact with each other?
Many use long whip-like structures called flagella for movement. They're quite different from eukaryotic flagella, though.
How so?
Bacterial flagella are more like rotating propellers. They're built from a single protein, flagellin, and they grow from the tip, pushing the bacterium along. Then you have shorter, sticker hair-like things called fimbriae. There are usually lots of these, and they mainly help with attachment sticking to food or surfaces.
Okay.
And then there are pili, generally longer, fewer per cell, and they have this fascinating role in conjugation.
That's the DNA sharing thing, right?
Exactly. Pili act sort of like grappling hooks. One cell extends a pilus, grabs another, and pulls them together so DNA can be transferred directly. And even more recently, scientists discovered these things called nanotubes, tiny direct channels connecting bacterial cells.
Nanotube, seriously?
Yeah. They seem to form direct conduits, allowing cells to exchange molecules from their cytoplasm, even between different species. It's like a real communication network down there.
Wow. Okay, so they have all these incredible structures and ways to interact, but how varied are their actual physical forms, and how do they make more of themselves?
Well, the oldest way we identify them is just by shape. You typically see rods, called bacilli, spheres, called cocci, or spirals, called spirilla. And while individual cells have these shapes, they often stick together after dividing. So you get chains or clusters or sometimes even quite complex colonies. Some, like actinomycetes, form long filaments. And myxobacteria can even aggregate to form these surprisingly complex structures called fruiting bodies, which contain resistant spores.
And we often encounter them as films too, don't we? Like on our teeth, you mentioned.
Exactly.
Oh.
Most prokaryotes growing on surfaces form what we call biofilms. These are complex communities, usually multiple species, all encased in this sticky matrix they secrete made of polysaccharides, proteins, DNA.
Like a tiny city built of slime.
That's a good way to think of it. And inside this biofilm city, the cells often communicate using chemical signals. It helps them survive better together and makes it easier to exchange genetic material.
Speaking of making more, reproduction. It's mostly binary fission, right? Just splitting in two.
Yes, primarily. It's a remarkably simple and efficient process. The cell copies its DNA, the two copies move apart as the cell gets longer, and then cytoskeleton-like elements help divide the cell down the middle into two identical daughter cells. Some also reproduce by budding, where a small outgrowth forms and pinches off, or by fragmentation of filaments.
So usually clones, but you mentioned adaptability.
Right. While binary fission produces clones, mutations are happening all the time. Combine that with their super fast generation times and you get incredible adaptability. A mutation that offers an advantage can spread through a population really quickly. But here's where it gets really interesting. Beyond mutation, prokaryotes have this powerful tool called horizontal or lateral gene transfer.
Right, sharing genes sideways, not just parent to offspring.
Precisely. It allows them to share genetic information directly between individuals, even ones that are only distantly related. It's a major driver of their evolution and adaptation.
And those mechanisms are the conjugation we talked about with pili, plus transformation and transduction.
Exactly. Conjugation is the direct transfer via pilus. Transformation is when a prokaryote just picks up naked DNA fragments directly from its environment, maybe from dead, plied cells.
Okay.
And transduction, that involves viruses, specifically bacteriophages, which are viruses that infect bacteria. Sometimes when a phage replicates, it accidentally packages up a piece of the host bacterium's DNA instead of its own.
Ah, so when it infects the next bacterium.
It injects that piece of bacterial DNA from the previous host. So the virus acts like a little delivery vehicle for bacterial genes. These processes, conjugation, transformation, transduction, are incredibly common in nature. They're constantly shuffling the genetic deck.
Amazing. And finally, for survival under really hard conditions, some bacteria have this remarkable trick, endospores.
Yes, endospores are fascinating. They're dormant, highly resistant resting cells. Certain bacteria, like Bacillus and Clostridium, form them, usually when things get tough, like when food runs out. They are incredibly resistant to heat, radiation, chemicals drying out, mainly because their internal protoplast is extremely dehydrated and protected by thick coat layers.
How resistant are we talking?
Well, to give you an idea, viable endospores have been recovered from 7,000-year-old lake sediments. And there are even reports, though maybe debated, of reviving spores from a bee trapped in amber, estimated to be 25 to 40 million years old.
Million? That's unbelievable survival.
It's an extraordinary survival mechanism, allowing them to wait out terrible conditions for potentially immense periods.
Okay, let's switch gears a bit. This metabolic diversity you mentioned earlier, how does that translate into their roles in the world? What do they actually do?
Prokaryotes are truly metabolic powerhouses. Their diversity here is just immense. Some are autotrophs, they self-feed.
Meaning they make their own food from simple stuff.
Exactly. From CO2. Photosynthetic autotrophs use light energy, like plants do, but others are chemosynthetic autotrophs. They get energy by oxidizing inorganic chemicals, things like nitrogen compounds, sulfur, or iron. Most prokaryotes, however, are heterotrophs. They need ready-made organic carbon sources, and a huge number of these are saprotrophs.
Saprotrophs. They feed on dead stuff.
Correct. They feed on dead organic matter. These are the great decomposers, the ultimate recyclers of the biosphere, working alongside fungi. Without them, nutrients would just stay locked up in dead organisms.
So what does all this metabolic activity mean for us, for the planet?
Well, their activities basically underpin life as we know it. They make huge contributions to the global carbon balance. They are uniquely responsible for nitrogen fixation, turning atmospheric nitrogen gas, which most organisms can't use, into ammonia, a form that plants can use. Only certain bacteria can do this.
That sounds fundamentally important.
It is. And through decomposition, they recycle all those essential nutrients, carbon, nitrogen, phosphorus, sulfur, making them available for new generations of life. Think about this. Over 90% of the CO2 produced naturally in the biosphere, so not counting human stuff, comes from bacterial and fungal metabolism.
Wow, and this raises an important question. You hear about bioremediation, using microbes to clean things up.
Yes, exactly. That leverages their diverse metabolic capabilities. Scientists are investigating and in some cases already using bacteria to break down toxic substances. Petroleum from oil spills, pesticides, industrial dyes helping to clean up pollution. Nature's own cleanup crew, in a way. Natural bacteria are already hard at work on oil spills, for instance.
Okay, so vital ecological roles, but they also impact us directly and not always in good ways. Disease, right?
Unfortunately, yes. Many significant human diseases are caused by bacteria, things like tuberculosis, cholera, typhoid fever. We even know now that bacteria, Helicobacter pylori, are linked to stomach ulcers. And in plants, they cause hundreds of highly destructive diseases, blights, soft rots, wilts, leading to huge crop losses worldwide.
But interestingly, not archaea. The chapter says no known diseases from them.
That's right. As far as we know, no archaea cause diseases in plants or animals. It's purely a bacterial domain issue, pathogen-wise. And those biofilms we mentioned, they pop up in human health problems too. For example, Pseudomonas aeruginosa forming biofilms in the lungs of cystic fibrosis patients makes infections really hard to treat. Or streptococcus biofilms on heart valves, or more commonly on teeth, causing gingivitis.
Right, the plaque on your teeth is a biofilm.
It absolutely is.
But it's not all bad news on the direct impact front. They're also commercially valuable, aren't they?
Oh, absolutely. Industrially, bacteria are incredibly important. They're the source of numerous essential antibiotics, like streptomycin and tetracycline, originally discovered from soil bacteria. They're used to produce drugs, vitamins, amino acids, industrial enzymes. And think about food production. Bacterial fermentation is key for cheese, yogurt, sauerkraut, pickles.
Couldn't live without cheese and yogurt.
Me neither.
No.
And looking forward, there's exciting work re-engineering bacteria like E. coli to produce biofuels. Plus, bacterial genes are widely used in genetic engineering, like the bait toxin gene from Bacillus thuringiensis, put into crops to make them pest-resistant.
Okay, so a huge range of impacts. Given this vastness, maybe we could highlight a few key bacterial groups mentioned in the chapter, starting with cyanobacteria.
Definitely. Cyanobacteria are incredibly important, both ecologically and evolutionarily. They perform oxygenic photosynthesis, like plants. They have chlorophyll A, carotenoids, and also these unique accessory pigments called phycobilins, which often give them their characteristic blue-green color, though some can be red or other colors. Their cells are usually packed with those internal photosynthetic membranes, the thylakoids. Many form slimy sheaths and can be single-celled, filamentous, or colonial.
And they're found everywhere.
Pretty much. From hot springs to Antarctic ice mats. And crucially, they were the organisms responsible for pumping oxygen into Earth's early atmosphere, a game-changer event. You can see fossil evidence of their ancient activity in layered formations called stromatolites.
Wow. And what's fascinating here, many also fix nitrogen.
Yes, a huge contribution. They convert atmospheric nitrogen gas into usable ammonia. This often happens in specialized, thick-walled cells called heterocysts, which protect the nitrogen-fixing enzymes from oxygen, which would inactivate them.
So they do photosynthesis and nitrogen fixation. How does that work?
Often, in filamentous forms, The photosynthetic cells provide sugars to the heterocysts, and the heterocysts provide fixed nitrogen back. It's a neat division of labor. This nitrogen fixation is vital. Free-living cyanobacteria in the oceans account for a huge chunk of nitrogen input, and symbiotic ones like Anabaena living with the water fern Azolla in rice paddies can provide all the nitrogen the rice needs. They can also form resistant restin cells called acinates, similar in function to endospores.
Okay, then the chapter mentions prochlorophytes, another photosynthetic group.
Yes, but unique. They contain chlorophylls A and B, just like plants and green algae plus carotenoids, but they notably lack those phycobilin pigments found in cyanobacteria. One genus, Prokaryotes, is famous for being the smallest known photosynthetic organism with the smallest genome.
Smallest but important.
Hugely important. It's thought to be the most numerous photosynthetic organism on Earth. Estimates suggest it makes up maybe 40–50% of the phytoplankton biomass in some ocean regions and could be responsible for producing up to half the oxygen generated on our planet.
Half the oxygen from this tiny thing. That's incredible.
It really puts their collective impact into perspective.
And the purple and green bacteria, how does their photosynthesis differ?
Their key difference is that they perform photosynthesis under anaerobic conditions, meaning without oxygen, and they do not produce oxygen as a byproduct.
So different chemistry involved.
Right. They use different types of chlorophylls, called bacteriochlorophylls, and have only one photosystem, unlike the two systems in cyanobacteria and plants. Their specific pigments give them their characteristic purple or green colors. Many use sulfur compounds, like hydrogen sulfide, H2S, the rotten egg smell gas, as their source of electrons for photosynthesis instead of water. This often leads to them accumulating granules of elemental sulfur inside their cells. You find them in sulfur-rich, oxygen-poor environments like stagnant water or sulfur springs.
Okay. Lastly, for bacteria, two more unique groups mentioned, mycoplasmas and phytoplasmas. What's special about them?
Their standout feature is that they completely lack cell walls. This makes them highly flexible, pleomorphic, meaning they can change shape, and contributes to them being among the smallest organisms capable of independent growth. They can basically squeeze through filters that would stop most bacteria.
No cell wall at all. Wow.
None. Mycoplasmas can be free-living saprotrophs or parasites of animals and plants. A notable group are the spiroplasmas, which are helical and motile without flagella, and they cause some nasty plant diseases like citrus stubborn disease. Phytoplasmas are very similar wall-less, tiny, obligate parasites of plants. They're responsible for over 200 distinct plant diseases, some really devastating ones like lethal yellowing of coconut palms. They live inside the phloem sieve tubes of plants and are typically spread by insects that feed on the phloem sap.
Fascinating. Okay, let's shift domains now to the Archaea. Once thought of as just extremophiles, relics living in harsh places, but the chapter suggests they're actually far more widespread.
That's absolutely right. While they are famous for thriving in extreme environments, high salt, high temperature, high acidity, we now know they're abundant in much more normal places too, like soil, and they're a major component of oceanic plankton, particularly the tiny picoplankton. Some estimates suggest they might even outnumber bacteria in the deep ocean.
And the key point, no known pathogens among them.
Correct. As far as we know, no archaea cause disease in humans, animals, or plants. A very important distinction from bacteria. What's fascinating here, we can generally divide them into three main physiological groups based on where they were first discovered, though we now know they're more diverse. First, the extreme halophiles.
The salt lovers.
Exactly. They thrive in incredibly salty environments like the Great Salt Lake, the Dead Sea, salt evaporation ponds. They actually require high salt concentrations, often 12–23%, for optimal growth. Their cellular components, like enzymes and ribosomes, are specifically adapted to function in high salt, stabilized by sodium ions. Some, like halobium, even have this amazing protein called bacteriorhodopsin. It's purple, and it acts like a light-driven proton pump.
Pumping protons with light.
Yeah. It allows them to generate ATP using light energy, but completely without chlorophyll or a standard photosynthetic pathway. It seems to supplement their energy production, especially when oxygen is limited in their high-salt homes.
Clever trick. Okay, second group, the methanogens, famous for producing methane.
Yes, methanogens are biochemically unique. They are the only prokaryotes known to produce methane gas, CH4, as a major metabolic product. And methane, as you know, is a potent greenhouse gas. They're also strict anaerobes. Oxygen is toxic to them.
So they live where there's no oxygen.
Exactly. Places like sewage treatment plants, swamps, bogs, deep ocean sediments, and famously in the digestive tracts of ruminant animals like cattle and sheep.
Ah, the cow burps.
That's them. They play a role in breaking down cellulose in the cow's gut, and the methane is a byproduct. A single cow can belch something like 50 liters of methane a day.
Wow. Okay, and the third main group, the heat lovers.
The extreme thermophiles, yes. These are the true heat lovers. Their optimal growth temperatures are typically above 80 degrees Celsius, that's 176 Fahrenheit. Some can grow at over 110 degrees C, even above the boiling point of water at sea level pressure.
How do they even survive that?
Their cellular components, especially their membranes and enzymes, are incredibly heat stable, with special adaptations to prevent denaturing or melting at those temperatures. Many of them metabolize sulfur, often using it as an electron acceptor in respiration, and most are strict anaerobes. You find them in hot, often sulfur-rich environments like volcanic hot springs, geysers, like in Yellowstone, and deep-sea hydrothermal vents, those black smokers. There's also a fourth, smaller group mentioned, represented by thermoplasma. It's unique because, like the mycoplasmas, it lacks a cell wall. It's been found pretty much exclusively in acidic, self-heating coal refuse piles. Another extremophile, just in a different way.
Incredible diversity in where life can exist. All right, moving on from cellular life now to viruses. These aren't even considered cells, really, but their impact on life is just undeniable.
That's right, they are fundamentally different. They're simple, submicroscopic parasites. An infectious virus particle, which we call a virion, is basically just a core of nucleic acid, their genome, which can be either DNA or RNA, single or double-stranded, all wrapped up in a protective protein coat called a capsid. Some viruses also have an outer lipid envelope derived from the host cell membrane.
And outside a host cell?
Metabolically inert, completely inactive. A virion is essentially just a delivery package. Its sole purpose is to get its genome into a living host cell and then hijack that cell's machinery, its enzymes, its ribosomes, its energy to make more copies of itself.
The ultimate parasite, in a way. And their impact on diseases, both in humans and plants, is just immense.
Indeed. They cause countless human diseases. Measles, mumps, influenza, the common cold, AIDS, COVID-19, the list goes on. And in plants, the chapter notes over 600 different types of viruses, causing more than 2,000 distinct diseases. The estimated global crop losses due to viruses are staggering, maybe around $15 billion annually.
What kind of symptoms do they cause in plants?
It varies hugely. It can be reduced growth, stunting, yield loss. Often you see distinct visual cues on the leaves, like mosaic patterns, which are patches of light green, yellow, or white intermixed with normal green, or maybe chlorotic yellow, or necrotic dead tissue ring spots. Some variegated flowers, the ones with patterned colors, are actually the result of a viral infection.
Huh, interesting. So how do these non-cellular things actually get into a plant cell and then spread?
That's a key difference from animal viruses. Plant cells have that rigid cell wall, which viruses generally can't penetrate on their own. Unlike animal viruses, which often bind to specific receptors on the cell surface to get in, plant viruses typically need help. Mechanical wounds are a major route damage from wind, handling, tools. But the most common way is through biological vectors, especially insects like aphids, leafhoppers, whiteflies that feed on plants. As they pierce the plant tissues with their mouthparts to feed, they transmit the virus. Viruses can also be transmitted through infected pollen, seeds, or vegetative propagation like grafting. Once inside a host cell, the virion disassembles it, sheds its protein coat, releasing its nucleic acid genome.
And then the hijacking begins.
Exactly. The viral genome essentially redirects the host cell's biosynthetic machinery. For many plant viruses, which often have positive-sense single-stranded RNA genomes, their RNA can act directly as messenger RNA, mRNA. The host ribosomes translate it to make viral proteins, including an enzyme, RNA replicase, that then copies the viral RNA to make more genomes. These newly synthesized viral proteins, the capsid subunits, then self-assemble around the new viral genomes to form progeny virions. Viruses come in various shapes. Common ones are icosahedral, which is a roughly spherical 20-sided structure, or helical, like the famous rod-shaped tobacco mosaic virus, TMV. In TMV, the RNA genome fits neatly into a groove formed by the helically arranged protein subunits.
Okay, so they replicate inside one cell. How do they spread to infect the rest of the plant?
Here's where it gets really interesting and connects to plant cell biology. They move from cell to cell through the plasmosmata.
Those little channels connecting plant cells.
Precisely. Now, normally, plasmodesmata are too small for a whole virion or even a viral genome to pass through. They have a size exclusion limit. But viruses have evolved special movement proteins. These proteins interact with the plasmodesmata and facilitate the passage of the viral genome, often as a complex with the movement protein itself.
So they basically unlock the door or widen the passageway.
Exactly. Some viruses actually modify the plasmodesmata quite dramatically, inserting a tubule through the pore that the virus then moves through. Others, like TMV, seem to just increase the pore's size exclusion limit significantly, maybe tenfold, allowing the viral complex to slip through. It's fascinating because studying how viruses manipulate plasmodesmata has actually taught us a lot about how these channels normally function in cell-to-cell communication in plants.
A side benefit of studying disease, I guess.
You could say that. This cell-to-cell movement through tissues can be relatively slow. But once a virus gets into the phloem, the plant's vascular tissue for transporting sugars, it can move much more rapidly throughout the plant, systemically infecting it, reaching distant leaves, roots, and growing points.
Makes sense. So how do plants fight back? They must have some defenses against these invaders.
Oh, they absolutely do.
Yeah.
Plants have evolved some sophisticated defense mechanisms. One is the hypersensitive response, or HR.
What's that involve?
This is often triggered when a plant has a specific resistance gene, our gene, that recognizes a specific component from the pathogen, and a virulence factor. This recognition triggers a rapid, localized cell death right at the infection site.
Sacrificing a few cells to save the plant.
Exactly. It creates a barrier of dead cells that physically contains the pathogen and prevents its spread. This is often accompanied by the production of antimicrobial compounds in the surrounding tissues. Then there's systemic acquired resistance, or SAR. This is a broader, more long-lasting defense.
How does SAR work?
It's typically triggered by an initial localized infection, like an HR response. This sends a signal throughout the plant. A key signaling molecule involved is salicylic acid, related to aspirin. This signal primes the rest of the plant, activating defense genes and making currently uninfected tissues more resistant to a wide range of subsequent infections, not just the original pathogen. It's like a plant-wide immune boost. And specifically against viruses, plants have a very effective mechanism called post-transcriptional gene silencing, or PTGS, sometimes just called RNA silencing or RNA interference, RNAi.
Gene silencing. That sounds powerful.
It is. The plant's machinery recognizes the viral RNA, often double-stranded RNA intermediates produced during viral replication, as foreign. It then chops this viral RNA into small pieces, which guide a protein complex to find and degrade other matching viral RNA molecules.
So it destroys the viral blueprints?
Essentially, yes. It targets the viral RNA for destruction, preventing it from being translated into viral proteins or used as a template for replication, effectively shutting down the viral infection or limiting its spread. It's a major antiviral defense in plants, and actually in many other eukaryotes too, including us.
Amazing defenses. Okay, finally, let's just briefly touch on viroids. The chapter mentions them as being even smaller and simpler than viruses.
Viroids are truly remarkable. They are the smallest known agents of infectious disease, period. They consist solely of a tiny circular molecule of single-stranded RNA. That's it. No protein coat, no capsid, nothing else.
Just RNA.
Just RNA. They range in size from about 246 to 399 nucleotides. That's much, much smaller than even the smallest viral genomes.
Yeah.
And crucially, they don't contain any genes that encode for proteins. They don't make anything themselves.
So how do they replicate or cause disease?
They are entirely dependent on the host cell's machinery. It seems they somehow trick the host cell's own enzymes, likely an RNA polymerase that normally works on DNA in the nucleus, into replicating their circular RNA genome. They cause disease symptoms not by producing viral proteins, but likely by interfering with the host cell's own gene regulation, perhaps by interacting with host RNAs or proteins involved in development or metabolism.
Incredible that just a tiny circle of RNA can do that. What are some examples?
The classic example is potato spindle tuber viroid, PSTVD, the first one discovered. It causes potatoes to become elongated, cracked, and, well, spindle-shaped, reducing yield significantly. Another devastating one is coconut kadang viroid, which has killed millions of coconut palms in the Philippines. They cause quite a range of diseases in various crop plants.
Wow. Okay, so wrapping this up, what does this all mean? From the sheer abundance and metabolic genius of prokaryotes to the ingenious non-cellular parasitism of viruses and viroids, it's clear that life at the microscopic level is far from simple.
Absolutely not simple.
These tiny entities, as laid out so clearly in this chapter, they really are the unseen architects and sometimes destructive forces shaping our entire planet, from global nutrient cycles to the diseases that challenge agriculture and our own health.
The depth of their metabolic diversity and their adaptability is just Truly astonishing. They are essential recyclers, the only nitrogen fixers, potential tools for biotechnology and cleaning up our messes, but also, yes, significant threats. Understanding them really is fundamental to understanding life itself and the interconnectedness of everything, really.
This deep dive has definitely shown us that even the smallest life forms, or maybe non-life forms in the case of viruses and viroids, have the biggest stories and the most profound impact. So here's something to think about. If these ancient, adaptable prokaryotes can thrive in conditions as extreme as boiling hot springs and deep-sea vents, places we can barely imagine life existing, and if viruses constantly evolve, adapting to every defense their hosts throw at them, what might this tell us about the ultimate resilience and the sheer evolutionary potential of life? Maybe even in the most unexpected corners of the universe. Something to ponder.